Hello, everyone. I am a speciation integration postdoc at UC Davis working with Graham Coop. I'm fascinated by animal diversity. Here are the species that I have worked with organized in the phylogenetic tree. I use biodiversity genomics to understand the fascinating irreversible lineage splitting events that generates new species. To replay the tape of life, I study animal species boundaries in the speciation continuum from the early stage to very late stage of speciation. This empowers my research program to understand the accumulation of key evolutionary events underlying different stages of speciation. Today, I will focus on these warbler species complex in the very early stage of speciation. Speciation is underpinned by the accumulation of genomic differences that are incompatible between species. Uncovering the evolutionary forces at these key genomic regions is critical for understanding speciation. The Pacific West Warblers is in the early stage of speciation with various layers of gene flow on the British Columbia coast all the way to Alaska. There's the ancient hybrid populations. And whenever the species come to secondary contact, there are hybrid zones. And there are even secondary hybrid zones formed between the ancient hybrid populations and the parental species. In this early speciation system, there are, despite the extensive gene flow in the rest of the genome, I have characterized this mitonuclear incompatibility that prevents gene flow. This is a gene block within chromosome 5 that harbors many mitochondria fatty acid metabolic genes that co-evolved with mitochondria. And so let's use the modern PubGen toolkits to reconstruct the evolutionary histories of these mitonuclear gene block. And basically, we took the whole genome sequencing of 5 individuals from the allopatric parental populations and reconstructed the ancestral recombination graphs with Argweaver. We tested whether there are signatures of long-standing barriers or species-specific sweeps. If we look at the coalescence trees of the randomly sequenced region in the genome, there should be paraphaly and random assortments of bifurcations. While if we sequence a region that serves as barriers to gene flow, there should be reciprocal monophyly. And if there are species-specific sweeps at certain genomic regions, we should see the recent time to the most common ancestor within species. And looking at this mitonuclear gene block into the time to the most common ancestor within species, we find this dramatic reduction in timing. Within both the sock in turquoise and stole in magenta. And relative to the genome size, randomly sequenced gene blocks of the similar sizes, there's significant reduction in this mitochondria nuclear co-functioning region, suggesting recent species-specific sweeps in both parental species. And the time to the last common ancestor between species can inform the long-standing barriers. Looking at this region, we find extended time to the most common ancestor in this gene block corresponding to such exaggerated coalescence trees where there's strong selective sweeps and deep coalescence time between species. And if we walk away from this region, the coalescence tree starts to become more random. And testing this signatures statistically, we compare the gene blocks of similar sizes in randomly sequenced regions of the genome, and there's indeed extended time to the most common ancestor in this mitochondria nuclear blocks. So this suggests both the species-specific sweeps and the long-standing barrier effect to gene flow in these mitochondria nuclear co-functioning blocks. From previous work, we find that this gene block, the genetic variation within this gene block co-varies with climatic variations. So such recent selective sweeps in both parental populations might be related to the climate change, both in the Pleistocene glacial cycles and the contemporary global warming. So we're looking into these possibilities now. And with that, I'd like to encourage any questions or comments, and please feel free to get in touch through Twitter if you'd like to. Thank you.